It starts the moment you take a bite too fast. A cold drink, a spoonful of ice cream, something frozen moving through your mouth before you've had time to think. And then, without warning, a sharp, spreading pain rises from somewhere behind your eyes and locks across your forehead. You press your hand to your head. You stop moving. You wait for it to pass. 15 seconds, maybe 20. Then it releases as suddenly as it arrived. Most people have felt this dozens of times. Almost nobody knows where it actually comes from. And here is the first thing that will surprise you. The cold never touched your brain. Not once. And there is something even more surprising than that. The brain itself has no pain receptors. The organ this experience is named after is physically incapable of feeling pain. So what is actually happening? And where is the pain really coming from? To understand brain freeze, you have to start somewhere unexpected: the roof of your mouth. Running along the upper palate is a network of blood vessels that sit unusually close to the surface of the tissue, closer than almost anywhere else in the head. When something very cold contacts this area rapidly, those blood vessels experience a sudden drop in temperature. The body responds immediately. Blood flow to the region increases, a warming response designed to restore normal temperature to tissue the body has detected as rapidly cooling. The blood vessels dilate. Warm blood rushes in. And this is where something unexpected happens. That sudden dilation is detected by a nerve that runs directly alongside those blood vessels, a branch of a nerve called the sphenopalatine ganglion, one of the largest nerve clusters in the head, sitting just behind the upper palate. When it detects that rapid vascular change, it sends a signal upward, toward the brain. But the brain misreads it. This is the part that makes brain freeze genuinely interesting. The sphenopalatine ganglion shares neural pathways with the sensory networks covering the forehead and the area around the eyes. When the signal from the palate travels upward, the brain, interpreting input through overlapping networks, registers the pain not where it originated, but where those shared pathways happen to map: the forehead, behind the eyes, across the temples. This is called referred pain. The experience of pain in a location different from the actual source of the signal. It is the same mechanism behind a well-known medical fact. Some people experiencing a heart attack feel pain in their left arm, not in their chest. The brain is not receiving incorrect information. It is reading the signal accurately. The signal itself is arriving through a pathway the brain associates with a different location. The pain is real. The location is a misattribution. Now, brain freeze is harmless. It passes. It leaves no trace. So why are neurologists paying attention to it? The answer is in the nerve at the center of all of this. Here is something that changes how you think about this. Brain freeze is one of the only examples of referred pain that is completely harmless, fully reproducible, and available to almost anyone with a cold drink, which is part of what makes it genuinely useful to researchers. Because the nerve at the center of this experience, the sphenopalatine ganglion, is not simply a curiosity of ice cream physiology. It is involved in some of the most severe and difficult to treat headache conditions known to medicine, including cluster headaches and certain forms of migraine. Some clinical research has explored whether blocking or modulating signals through this ganglion could help manage these conditions. Brain freeze, in that context, is not simply an inconvenience. It is an accessible, reproducible model of a pain pathway that researchers can observe without causing lasting harm. The medical term for it, sphenopalatine ganglioneuralgia, reflects exactly this: pain generated specifically through that ganglion. Every time brain freeze is triggered, the same nerve cluster implicated in some of the most debilitating headache disorders is briefly and harmlessly activated. And here is what makes this particularly significant. Unlike most pain pathways in the body, brain freeze can be triggered deliberately, observed in real time, and studied as it resolves, all without causing any lasting harm to the subject. Researchers studying headache disorders rarely have access to a pain model this clean. Most pain is unpredictable, inconsistent, or impossible to induce safely. Brain freeze is none of those things. It is one of the few pain experiences in medicine that arrives on schedule, behaves consistently across trials, and disappears without a trace. That predictability, trivial as it seems in the middle of eating ice cream, has genuine scientific value. This raises a question most people never think to ask. Why does brain freeze stop? The temperature at the palate begins to normalize as the body's warming response succeeds. Blood flow stabilizes, the vascular change that triggered the ganglion subsides. The signal diminishes, the referred pain fades. The whole episode, from trigger to resolution, typically lasts between 20 and 30 seconds, though duration varies between individuals. Now, you may have heard that pressing your tongue firmly to the roof of your mouth can shorten a brain freeze episode. The biological reasoning is plausible. The tongue is warm, and contact with the palate may help restore temperature to the affected area more quickly, potentially reducing the duration of the vascular change driving the signal. Current evidence supporting this as a reliable intervention is limited, but the logic is consistent with the known mechanism. Why doesn't everyone experience brain freeze the same way? Not everyone experiences it with the same intensity, or at all. Current evidence suggests that differences in the anatomy of the sphenopalatine ganglion, the density of blood vessels in the palate, and individual sensitivity in the relevant neural pathways may all contribute to why some people are highly susceptible while others rarely notice any pain. And here is a connection that researchers find significant. People who experience migraines appear in some research to be more susceptible to brain freeze, a finding consistent with the involvement of shared neural pathways in both experiences. The exact relationship between brain freeze susceptibility and migraine remains an area of ongoing investigation. But the pattern suggests that what feels like a trivial response to cold food may reflect something deeper about how an individual's pain signaling networks are calibrated. Return for a moment to where this began. The cold never reached the brain, and the brain has no pain receptors to feel it with. What you experience as brain freeze is generated by a nerve cluster in the roof of your mouth, referred through overlapping neural pathways to the surface of your head, and placed by a brain reading the only architecture it has at an address that doesn't match the origin. It is a precise, involuntary demonstration of something worth understanding, that the brain can be confidently wrong about where something hurts, and that the location of a pain and the source of that pain are not always the same thing. And so, the next time you take a bite too fast, the cold moves through your mouth, the pain rises behind your eyes, you press your hand to your forehead and wait for something to pass that never originated there in the first place. 15 seconds, maybe 20. Then it releases, the same way it always has. And now you know why. What felt like your brain freezing was actually your nervous system revealing one of its oldest shortcuts. If you're curious about how your mind and our world really works, subscribe to WhyDecoded, because everything has a reason. We just haven't decoded all of them yet.